Okay, should we wrap up our discussion on nucleotides? Are there any questions with the purines? So we'll return to the purines when we discuss disorders of nucleotide metabolism. But before we do that, we'll talk about the pyrimidines. So pyrimidine metabolism is quite simpler. I mean, their structure is simpler, their synthesis is simpler, and they don't have as many disorders of metabolism in them. So pretty straightforward. Okay, so the pyrimidine bases are uracil for RNA and cytosine and thymine for DNA. The common intermediate is uracil, or well, first orotate as a base and then uracil as the nucleoside. The synthesis of the pyrimidines, again, in contrast with the purines where you start with a PRPP molecule, the pyrimidine synthesis starts with an enzyme that is, kind of familiar from urea cycle discussion, carbamoyl phosphate synthase. This is a different carbamoyl phosphate synthase, though. This is CPS2, and carbamoyl phosphate synthase 2 does not use inorganic ammonium as a nitrogen donor. It's using the amino acid glutamine. Okay, so this is CPS1 is one of the three enzymes that can fix free ammonium. CPS2 is not. It makes the same product, carbamoyl phosphate, and it's using carbon dioxide and ATP like CPS1, but it's not using ammonium, it's using glutamine. Okay, so this carbamoyl phosphate then, this binds with aspartate, and that's basically the structure of that first ring of the single ring of the pyrimidine base. So this can get modified down to orotate. The orotate base then, this gets transferred onto the ribose sugar using that same phosphoribosylpyrophosphate, pyrophosphate, and the product then is the uridine monophosphate nucleotide. So that's shown in a little bit of detail here. Okay, so, or a little bit more detail here. So we've got the aspartate joining with carbamoyl phosphate, and essentially that's all the atoms that we need to make the pyrimidine base. We get ring closure reactions to make the orotic acid or orotate. Then the the phosphoribosyl pyrophosphate is added to make the orotate nucleotide. Then this acid portion is cut off as carbon dioxide to make the uridine monophosphate. Okay, so this pathway, if you recall from the urea cycle, this is where that carbamoyl phosphate from the urea cycle can impinge on the pyrimidine synthesis pathway and result in orotic acid increase. Okay, so if we have, for example, an OTC deficiency, carbamoyl phosphate from the mitochondrial urea cycle spills out into the cytosol and can be a substrate for pyrimidine synthesis. If you remember from the beginning of the last hour, the phosphoribosyl pyrophosphate synthesis, that's a major regulatory step in nucleotide homeostasis. So in a cell that is not actively replicating its genome, for example, or it doesn't have a need for nucleotides, it's not going to be synthesizing PRPP, so the pathway will stop at this point, and orotic acid will build up and then be urinated out. Is that helpful from the linking that with urea cycle? Okay. Okay, so we now have uridine monophosphate, and what happens to the molecule next is going to depend on whether that molecule is going into DNA or going into RNA. So I'll do the RNA first because that's a little bit simpler. So with RNA, uridine monophosphate can get phosphorylated twice, thrice to uridine triphosphate, and that then can be a substrate for RNA polymerase to incorporate into RNA. The uridine triphosphate, this can be aminated by using glutamine as a nitrogen donor to make the cytosine triphosphate. This then is ready to go into RNA. Okay, so to make the deoxypyrimidine bases for DNA synthesis, we have to go through a little bit more circuitous route. And that is shown here. So the rationale for that is the thymidine synthase, that enzyme that we've seen a few times, that is using deoxyuridine monophosphate as a substrate. Okay, so we know that the ribonucleotide reductase always uses a diphosphate form, right? So if we want to get to thymidine synthase from UDP, we take the diphosphate form to make deoxyuridine monophosphate, then a phosphatase clips off one of these phosphates to make deoxyuridine monophosphate. Deoxyuridine monophosphate, then this is the substrate for the methylation by thymidine synthase to make deoxythymidine monophosphate. Deoxythymidine monophosphate, this is phosphorylated twice, thrice to make deoxythymidine triphosphate, which is a substrate for DNA polymerase. Okay. Let's see, to make the cytidine triphosphate nucleotide, in this case, the cytosine triphosphate, this has to be dephosphorylated to make cytosine diphosphate. The diphosphate is a substrate for ribonucleotide reductase, so that makes deoxycytosine diphosphate. This then can be phosphorylated to the triphosphate form to make deoxycytosine triphosphate, which is a substrate for DNA synthase. Let's see, so we can also get conversion of the deoxycytosine diphosphate into the DUMP through a dephosphorylation and a deamination reaction. Okay, so this step is important pharmacologically because cancer cells need to do a lot of DNA synthesis, right? So that's the rationale for targeting this pathway with drugs like pro-drugs like 5-fluorouracil that can block this pathway and deplete the cancer cells of the necessary substrates for DNA synthesis. Okay. So with the degradation and the salvage with the pyrimidines, it's a lot simpler process. So we don't have the conveyor belts of the salvage pathway, and we don't have the issue with the buildup of uric acid as a potentially pathological intermediate in the pyrimidine salvage. So the pyrimidines are degraded into compounds that can either be urinated out or be used as energy. So the beta-alanine, this is like alanine except with a amino acid switched out. This can be urinated out. The beta-amino isobutyrate, this can be converted into butyric acid, and then that can be converted into the methylmalonic acid and succinate through that pathway that depends on biotinylation and, biotin carboxylation and vitamin B12 dependent rearrangement. Okay, so pyrimidine metabolism is quite simple compared to purine metabolism. Okay, should we do one review question. And right, look how easy that question is to write. I could write that question in 6 different ways in about 2 minutes, right? Okay. Okay, so make sure that you don't get that one wrong. Okay, ready? So ribonucleotide reductase always uses a diphosphate form of a nucleotide. Okay, its substrate is always going to be a nucleotide, and its product is always going to be a deoxynucleotide. Okay, so cytosine diphosphate would be a substrate for ribonucleotide reductase. What if what?
You just say what you said just one more time.
Yes, ribonucleotide reductase is always using a diphosphate nucleotide as a substrate. And its product is going to be a deoxynucleotide that is diphosphorylated. Good? Okay. So best answer here is cytosine diphosphate. Now let's consider answer A. What if answer A was deoxyuridine diphosphate? Would that make it a correct answer? No? Why not? Right. Good, because it's already a deoxy. And a deoxynucleotide, that's a product of ribonucleotide reductase, not a substrate. Everybody clear on that? Okay. All right, so let's spend the remainder of our time talking about disorders of nucleotide metabolism. Most of these are occurring in the purine nucleotide metabolism pathway. There's one condition, PRPP synthetase superactivity disorder, that's affecting kind of, upstream of either pyrimidines or purines. So we'll talk about that one first. So the PRPP synthetase, this enzyme is normally allosterically inhibited by ADP and GDP. So the diphosphate products of the purine nucleotide synthesis are inhibiting the activity of PRPP synthetase. So this is an example of feedback inhibition of the nucleotide biosynthesis pathway. There is an inborn error of metabolism in which that feedback inhibition is lost due to inherited mutations in the PRPP synthetase gene. So this results in PRPP synthetase superactivity disorder. And this is interesting because it's an inborn error of metabolism that results in overactivation of an enzyme, where generally mutations are resulting in inactivation of mutations. The overactivation of the enzyme results from losing that allosteric inhibition. Okay, so the symptoms of the PRPP synthetase superactivity disorder result from unregulated synthesis of the nucleotides. Their unregulated synthesis results in increased breakdown of the nucleotides, and you can get a hyperuricemia, so increased uric acid as a result, and that can manifest as either gout or gout arthritis or stones in the kidneys or the urinary tract. There can be much more severe forms that will manifest in a young infant and result in more severe neurodevelopmental problems, and the pathophysiology of those are beyond the scope of this class. So that PRPP synthetase superactivity disorder, again, that's resulting from that PRPP synthetase losing the feedback inhibition from the purine diphosphates, the ADP and GDP nucleotides. Okay, so the PRPP synthetase, that's one of the major levels of regulation of the nucleotide synthesis pathway. Okay. Questions about PRPP synthetase superactivity disorder? Okay. So we're basically getting too much production of these nucleotides, and the degradation of the purine nucleotides is going to result in uric acid, which can build up and cause the problems. Now, presumably, we're also going to get increased synthesis of the pyrimidines as well, but they don't have those issues with the toxic or potentially toxic degradation products. Okay, so let's look at a question related to this. Okay, so gout and kidney stones, wouldn't be abnormal in a 60-year-old, I guess, or wouldn't be so unusual, but in a 1-year-old, that's definitely not normal. And best answer, good, PRPP synthetase disorder, or PRPP synthetase superactivity disorder. Okay, should we look at another case? All right, so I think all of the remaining cases are in the purine salvage pathway. So the first case, we have a 2-month-old male with fever, sore throat, diarrhea that was refractory to antibiotics. We have, let's see, very low levels of lymphocytes and some indication of liver damage. Let's see. High protein in the cerebrospinal fluid and low glucose in the cerebrospinal fluid. Did Dr. Diebel talk about CSF infections and glucose and protein in the cerebrospinal fluid? Okay. That's a consequence of meningitis. Okay, so an infection. But there was no culturable organism here. Okay, so the provisional diagnosis was meningitis, and there was intravenous antibiotics were started. And 4 days later, he started to get a lot worse. So the chest x-ray, I'm going to leave this to radiologists, but there's supposed to be a thymus somewhere in the neck. I imagine that's where the thymus is, that you can see on x-ray, which is not present in our case. There's also more haziness in the lungs than normal, and I'm just reading what's described there. You're going to get chest x-ray interpretation soon. Okay, so we've got a pneumonia, and again, it was this, here they're calling it pneumocystis carnii, but this has now been renamed a decade ago or so as pneumocystis jirovecii. Okay, so what's our picture? We've got a child with below normal levels of lymphocytes with pneumonia that is caused by an organism that only causes pneumonia in people that are severely immunocompromised. How would you handle this patient then? Parenteral folate. Right, same, very similar condition to that other proton-coupled folate transporter deficiency, right? Okay. This is a different case, though. So we've got T cells and B cells and natural killer cells, so these are all greatly decreased. The red blood cell adenosine deaminase enzyme activity was absent. And genetic analysis revealed that there was a homozygous mutation in the gene, in the adenosine deaminase gene, and that same mutation was found in both of the parents. They were heterozygotes. This is a cytosine to thymine mutation. So this is A to V, alanine to valine. Is that a conservative substitution or a non-conservative substitution? Did Cormier talk about mutations and conservative versus non-conservative mutations? Okay, what do you think of alanine to a valine?
I mean, they're both hydrophobic, but one's branched, so I don't know. It's probably not that big of a deal, but apparently in this case it is.
Yeah, this would be a non-conservative because of the bulkiness of the valine. That's gonna be a non-conservative. Chemically, they're still gonna be hydrophobic, but I think just the bulk of that branch chain, side chain, is gonna be make it non-conservative. Okay, so this patient was given a hematopoietic stem cell transplant, and there was a matched sibling, so an HLA matched sibling who did not inherit the same ADA mutation. So the HLA matching and graft versus host disease, you're going to get that in spring semester. So we'll just kind of skip over that. 18 months later, they did a second one. And this is the profile of the lymphocytes. So before the hematopoietic stem cell was here, so they were tanking. Then they were increasing, but kind of plateauing here, and then there was a second hematopoietic stem cell transplant, and that resulted in more sustained lymphocyte proliferation. Okay. So the T cells are the more abundant lymphocytes. The B cells didn't go up a lot, but at least the B cells are going to be functioning because B cells require T cells in order to function. So the pathology in this case, we have a deficiency in the adenosine deaminase. So that's that salvage pathway enzyme that is removing the ribose removing the amine group from the adenosine as part of that salvage pathway. So the toxicity here, or the pathology, comes from an accumulation of adenosine and deoxyadenosine, and this buildup of adenosine and deoxyadenosine, this is toxic and specifically toxic to developing lymphocytes. So as lymphocytes are, trying to populate the thymus and trying to mature, they die instead. Okay, so that enzyme is here, adenosine deaminase, in our salvage pathway. And it shows the importance of the salvage pathway. I guess you would think that this wouldn't be, there wouldn't be enough flow through here to allow adenosine to build up, but there is. Let's see. So the main toxicity is that buildup of adenosine and deoxyadenosine, and that's toxic to the developing lymphocytes. Let's see, so the symptoms are the low lymphocytes, and then this costochondral junction dysplasia. I'm not sure if you can see that, but I think it's this arrowhead. So the costochondral junction, that's the junction between the bone and the cartilage portion of the rib. And that can get all inflamed, and I guess I'm not sure how to see that, but I think it's probably these arrowheads. Okay, I'm not a radiologist, so. Okay, so the treatment then is bone marrow transplant. So try to identify an HLA matched donor that doesn't carry this mutation. Then the bone marrow precursors of those lymphocytes have healthy adenosine deaminase, and they're not subject to that adenosine toxicity as they're developing. Does that sound good? Okay, so this is a condition called adenosine deaminase severe combined immunodeficiency. Did Dr. Scroggins talk about immunodeficiencies at all? Like IL-2 receptor? Or did you talk about the interleukin-2 gene or IL-2 receptor at all? No, okay. That's the more common cause of a severe combined immunodeficiency is deficiencies in those genes. But you'll get a lot more of this in spring semester. Okay, so let's look at another case. So this is combined immunodeficiency, and the clinical path of this case was worse than the previous case, but this is generally considered to be a less severe condition. And we can kind of see that. So the first one was a few months old kid, and this is a 7-year-old girl. So that's it's a slower burn for the pathology. Okay, but she has a history of respiratory tract infections. And yeah, so kind of a lot of negative stuff. Boy. I'm just drawing a blank on these musculoskeletal abnormalities, but if anybody wants to Google that, go for it. Okay, so let's look at some of our laboratory values. So she's anemic. We don't have a mean corpuscular volume for this patient, so we can't tell whether it's microcytic or macrocytic. Leukocytes are down, and the neutrophils and the total lymphocytes are down as well. So one thing, of course, that wasn't done in our previous case of immunodeficiency, but anytime you've got recurrence infections, and especially opportunistic infections, Pneumocystis jirovecii or oral thrush or things that don't infect people with healthy immune systems, HIV test is, that's easy to do and should be done in that case if you suspect an immunodeficiency. But it was negative in our case. Okay, so if we look at some of the more detail about the hematological profile, so the CD, these are referring to clusters of differentiation. And have you heard of these before? Or okay, so they're proteins that are transmembrane proteins, and you can detect them on the surface of cells. And they just, when they didn't know what the function of those proteins were, they just started numbering them, CD1, 2, 3, 4, etc. Right? And then they found out what they were for, and they kept the CD numbers. Okay, so CD3, this is a T cell marker. And the mature T cells are either CD3, CD4 positive, or CD3, CD8 positive. So these are the two families of the T cells, and they're both decreased in number. As are the B cells. So B cells, the CD19 and CD20, these are B cell markers, and these are both decreased. If you compare the numbers to the adenosine deaminase severe combined immunodeficiency, they're not as bad. So they're not going down to 0. We looked at the adenosine deaminase activity, and that is in normal ranges within a normal range. But the purine nucleoside phosphorylase activity, that is greatly reduced. Okay. So in this case, she would be the child would be a candidate for a hematopoietic stem cell transplant, but unfortunately there was not a donor that could be identified. And the patient succumbed to the fungal infection of the lungs, and the fungal infection of the lungs by a agent that, normally if you had a healthy immune system, you wouldn't ever get that infection. Okay, so this is a case of purine nucleoside phosphorylase deficiency or purine nucleoside phosphorylase combined immunodeficiency. So in this case, the pathology is similar. You get a buildup of adenosine nucleosides because you can't go through this salvage pathway to make hypoxanthine. So that adenosine nucleoside and deoxyadenosine, that is toxic to the developing lymphocytes, and if they're not around, then the patient can be susceptible to these opportunistic infections. So the diagnosis, the distinguishing like purine nucleoside phosphorylase deficiency from ADA deficiency, that's really going to be in the purview of the immunologists and hematologists. So that's not something that I'm going to ask you all to do, unless you're given data like this, okay? If you're given these two activities, you should be able to distinguish between PNP and ADA CID, okay? But just looking at the lymphocyte levels or what opportunistic infections they are, that's going to be beyond this, your scope, okay? But generally, the PNP phosphorylase deficiency, the lymphocytes aren't as low as they are in the ADA deficiency. Okay, so the PNP phosphorylase deficiency is considered to be a less severe combined immunodeficiency. However, as we see in this case, it can still have fatal consequences to the people who have it. Okay, are there questions about these two forms of immunodeficiency? Okay, let's move on and talk about, I think this is our penultimate disorder, and it's related to the ultimate one, Lesch-Nyhan syndrome or Lesch-Nyhan disease. This is a rare condition, X-linked, so it's manifesting in males, and this is caused by inherited mutations that eliminate the function of the hypoxanthine guanosine phosphoribosyl transferase gene. So the condition is manifested as hyperuricemia. So this can be have the conditions of gout or kidney stones. There can also be such extreme hyperuricemia that you're getting like uric acid crystals in the diapers of the child who has it. So they're peeing out a lot of uric acid. The kind of thing that would distinguish this from just a simple gout is the really profound cognitive delays and behavioral disorders that are pretty peculiar to this this condition. There is a self-injurious behavior and a biting the lips and biting the fingers, and it can be, kind of pretty severe, as shown in this finger, resulting in loss of the fingers and having the patient have to be restrained for much of his life. So this, human behavior is complicated, certainly, and because this is a very rare condition, It's hard to get the exact pathophysiology about why this happens, but there are some cell culture models and animal models that can kind of recapitulate some of the behavioral features of Lesch-Nyhan, and the dopamine signaling in the central nervous system is implicated in that kind of odd self-injurious behavior. Let's see, what else was I gonna? Oh, there's also, let's see, dystonia, or just kind of difficult movements of the limbs is another common feature. Let's see, allopurinol. Allopurinol is an inhibitor of xanthine oxidase. So the xanthine oxidase, that's that enzyme that is making uric acid, if you remember. Right? So the idea with the allopurinol treatments is to slow the generation of uric acid to a rate that can be urinated out without the generation of the kidney stones or the gout. So the allopurinol treatment, this can be effective in resolving the hyperuricemia in patients with Lesch-Nyhan, but it doesn't have any effect on the neurological self-injurious behavior or with the dystonia or mental retardation. Okay, so this one, so this is an X-linked condition. Right, so it only manifests in males. The enzyme that is deficient is hypoxanthine guanine phosphorybosyl transferase. Right, so you can remember that by he's got purine recycling trouble. Right, so that tells you it's a he, so it's X-linked. And then it tells you exactly what enzyme it is. Right? Like that? No charge. Okay. Okay, so this is an example of a question that we could you could use to assess your understanding of this concept. Okay, everybody got a chance to answer this one? Okay, good. So B, transferring PRPP to guanine or hypoxanthine, that's the enzymatic activity of the hypoxanthine guanine phosphorybosyl transferase enzyme that is deficient in Lesch-Nyhan. Okay, any questions about Lesch-Nyhan? Okay. Let's look at this question. Okay, ready to look at the answer here? Okay, good. Yep, a suicide inhibitor of xanthine oxidase. That's the best answer. So the condition here is gout, right? And this is where linked to the case where that came from, and this is what it would look like on a radiograph. So this little abnormality here, that's composed of crystals of uric acid. Okay, and as you can imagine, it would be quite painful, I guess, if you had that in your finger. So the uric acid, that is a result of nucleotide catabolism, of the purine nucleotide catabolism. And the therapeutic goal is basically to slow the production of uric acid to a level that you can urinate out without it precipitating and forming that crystal. Okay, so allopurinol, this acts as a inhibitor of the xanthine oxidase enzyme. It acts as a suicide inhibitor. It acts as a substrate of the enzyme. So the allopurinol itself is modified by xanthine oxidase, and it's made into a product. I think it's called hydroxypurinol, but I don't know if I've got that written here. Yeah, hydroxypurinol. That product then is so tightly bound to the xanthine oxidase enzyme that it can't be competed away. So it's not a covalent bond, but it's bound so tightly that it's effectively, the kinetics of it are acting like a suicide inhibitor. Okay, any questions about gout then? So this would be an example of like idiopathic gout, or gout that doesn't result from a inborn error of metabolism, but just is a unfortunate consequence of aging. Any occupational contribution to this gout? Well, lobster fisherman is probably going to be putting his hands in cold water a lot, right? Because I think lobsters are mostly in like northern Atlantic, not the warm waters of the Mediterranean. So that cold water, the solubility of this uric acid, whether it's a solid or whether it's soluble in urine or blood, that's going to be dependent on temperature. So the colder something is, the more likely it is to be insoluble and to precipitate out into a crystal. So that's why these tophus, these uric acid crystals, they happen in the extremities and not in the shoulders or the vertebrae. Okay. Yeah. That's all I've got for you. Yeah, oh yeah, you have a question?
So when you're giving one of these drugs that basically, with the goal of preventing uric acid buildup, is there any side effect of the increased substrate concentration, have you?
I don't think so. I think that these upstream things are not pathologic. I mean, if you get up to the level of adenosine and deoxyadenosine, then that could be problematic, as we saw with the cases of the combined immunodeficiencies. But, the development of the uric acid is a few steps away from that. So, yeah, good question though. Any other questions? Okay. All right, I'll load up the quiz slides shortly. All right, thanks.
What? Yeah. Oh, thanks. Yeah.
That was a bad. Yeah, that was like that was bad.
Yeah, it's kind of fireside.
I haven't edited it in 6 hours since last time.
And Dr. House. She had something last night. I told her.
I didn't know you had the art. You gave us something. It was just on last night.
I heard one of the LCs just like.
Serving 30 minutes. Right? It worked.
Dr. Hart. Dr. Hart used to rocks.
Damn. You lucked out.
Yeah, and
Let's walk for one then.
Yeah.
Exactly.
I shared quite here.
Appreciate the class.
Yeah, you, me, and Kai have an alliance.
I love it.
Kai's all about clapping too.
Yeah.
Especially today, he's like I was dying.
Oh, I checked out. Easily. Oh my gosh, that's awesome.
You brought up to your guys' house? Yeah.
Awesome, I'm so jealous.
I know, it like makes me so happy when I like walk downstairs.
Oh, my plants make me so happy, so I can definitely relate, but that's nice. Plants are expensive.
They are.
Yeah.
So good. I know, but plants like that big would be ridiculous.
Hundreds, yeah. That's why I'm trying to,
Honestly,
on
if you go to
Facebook
Facebook
Marketplace.
Marketplace, yeah,
I was just gonna
people who are moving are always trying to
yes,
get
get rid of their plants, yeah.
Really?
Do that more.
I should find something.
My mom has been like giving me an obscene amount of plants.
Really?
Because she like has so many during the summertime.
Yeah.
She puts them all at like noon, and then filled up my entire car with a bunch of plants that I'll probably kill.
We are springtime now.